Hello, good evening, friend. Today I am going to explain an important topic of surface chemistry, and the topic is mechanism of enzyme catalysis. Okay, in the few video, you know, uh, previous to this video, I also explained the mechanism of the catalysis of you know catalyst. So they were, that is, firstly the reactant get adsorbed on the surface of catalyst, then they form intermediate, and then they get deadsorbed and form the product. Now, what is the mechanism of the enzyme catalysis? Okay, so case of enzyme is quite little more specific compared to the that one catalyst for any gaseous molecule can get a job on the surface of the catalyst. But in case of enzyme it is not like that. It is a very much specific, the which reactant will fit on the enzyme surface, that will only react, other will not. So let us see, firstly, background what could be. If suppose there are the enzymes are there, on the enzyme surface, enzyme surface, there are there are there are you know uh, cavity present. There are cavities, with with fixed shape present. Also they have you know they have functional group. Also they have functional group functional group such as you know NH2, C double OH, SH, OH. It's any group maybe, depending on the nature of catalyst. And what they do, the the molecule of reactant, the molecules of reactant, the molecules of reactants, the molecules of reactants, and either you can write molecule of reactants or substrate, you know substrate in bracket S, which have you know which have complementary, complementary complementary shape, to cavity, you know to cavity of enzymes, fit into this cavity, fit into these cavities, fit into these cavities, just like what, just like you know just like just like uh, key fit into lock, key fit into lock key fit into lock. Okay, you know the this one, the mechanism involved two step. The mechanism mechanism mechanisms involves following two step, following two steps. Now let us see what are the following two steps. Okay, first one is step number 2, is step number 1. What is step number? I know Firstly, what the reactant will bind on the surface of catalyst. So how can we write? Binding of binding of substrate, you know bracket S, or reactant, both are the same thing, or reactants, to the active site of enzyme, of enzyme active site of enzyme, in bracket cavities. Okay, to form the activated, what to form to form the activated to form the activated enzyme enzyme substrate enzyme substrate complex. enzyme substrate complex, that is ES. Okay, now let us see with the diagram how can we understand. We have the substrate like this. Okay, firstly we will draw the enzymes, then we will be able to understand, because a particular important thing is enzyme. Okay, so enzyme has the cavity like this. Okay, these are the cavity having the some functional group and fix, shape. What will happen at the same time, we have the we have the substrate also. And what the substrate is there, this substrate is capable having also the capable to fit into the active site of the enzyme. So these are what, these are the cavities, you know cavities with a specific shape, you know specific shape. Okay, only that reactant will, this is the substrate, only that reactant or substrate will fit, which have the complementary shape of these cavity. I know this is the enzymes. Okay, enzymes. So enzymes is denoted by E and substrate is S. So what will happen? Okay, what will happen, this will be, next step, this step they will fit completely. Okay, and what kind of structure they will form, that we have to analyze. Okay, so these are the structure. In this one, first one is the fitting in this cavity, then another cavity, and then another cavity. So if you see, there were a little line will be there. In this line, what has happened, there is the substrate is fitting. Okay, there is the little darker line where there is the substrate and catalyst are fitting each other and forming the enzyme substrate, and forming the enzyme substrate complex. So this is known as what, ES complex. ES complex. Okay, so now we'll move for the next step, and we'll write the next step, then we do the diagrammatic. Step number 2 is step 2. So what is step 2? Formation of product by decomposition of the activated enzyme substrate complex. In this step, in this step, enzyme, in this step, substrate enzyme substrate in bracket ES, enzyme substrate complex, enzyme substrate complex decomposed, enzyme substrate complex decomposed, and decomposed into the decomposed into the products. Okay, products. Let us see what are the products, how the diagrammatically how we can explain the decomposition of enzyme substrate complex. So earlier what was there, the earlier if you see what was that, the ES complex. Firstly I am doing the ES complex, then only will be able to do another one. This was the ES complex, enzyme substrate complex, and these complex are going decomposition. Okay, in the decomposition in the next step. Okay, so what will happen in the next step, that will form the 3 products. Okay, 3 cavities are there, so according to the cavities they will form. So enzyme will be the, as it is at the end of the reaction, and what will be the enzyme, as the cavity will be there on the surface of the enzyme. Okay, so this is the enzyme. Okay, enzyme, there is no change on the enzyme. Whereas if you see the other part of the reaction, okay, so first one is the P1, product number 1, and then one more product will form here. This is also having the similar cavity, similar shape of fitting with the cavity of the catalyst. So this is P1, this is P2 and P3. So what we are going here, this is enzyme. So finally P1, P2 and P3, they are the P3 are the products. P3 are the products. So these were the catalyst enzyme, enzyme mechanism. So what we study in this lecture, that on the surface of the enzyme there is the functional group, and along with that there is a cavity, and the cavity is in the fixate. Okay, only that reactant will get react which have the complementary shape to fit in the cavity. How they fit? Just like the key fit into the lock. So firstly, in the there's the mechanism involved into two steps. In the first step, substrate fit into the cavity of the enzymes, okay, and form the enzyme substrate activated complex. And very next step, the whole they go under the decomposition reaction, and enzyme substrate complex, what are the activated enzyme substrate complex, deactivate and give the products and enzymes. So these were the mechanism of the reactions. Thank you so much for watching, friends.